We will be recording, and I'm starting the recording now. Yes, so this webinar will be recorded. So as I said, this is the Plants for the Future European Technology Platform webinar on genome editing, and we're going to discuss the impacts on the transition towards more sustainable agriculture in Europe, and how gene editing can play a role in this. And basically, this is an interactive webinar. We will be using Mentimeter. So basically, we ask that you go to menti.com, as you can see here, and use the code provided. It will be provided throughout the slides, but please connect as soon as possible. And we have a few questions for you, and this will also be the way that we will ask questions ourselves. Please do not use the chat for questions, only ask questions through the Mentimeter. So before I start the introduction, I will just give some brief instructions. So obviously, please stay muted and turn off your cameras during the webinar if you are not presenting. And as I said, the webinar will be recorded. And again, please go to menti.com and use this code so that you can ask questions to the panelists and the speakers. You can also upvote each other's questions, and you can also answer our questions. So the way it would look, for example, if you're using a smartphone, you would have, for example, the questions that we would be asking at the moment that you can answer. You can then press submit, but you can also actually go to this gray button that's ask a question, where you will be brought to another page where then you can ask your questions. You will be able to see each other's questions, and if you want to upvote a question, you press this thumbs up. We will obviously not be able to answer all the questions. That's why we would like to use this voting system, so that we can address the questions that you think are most relevant. So the first question that we have actually for you is to get an idea of our audience profile. We'd like to ask what the stakeholder group you represent. So if you go on menti.com and you use this code, then you will have the options there, and we'll look at the results of that after my introduction. So just briefly, the agenda for today. I will then, as I said, do a quick introduction. Then I will leave the floor to Professor Holger Puchta, who will talk a little bit about the technology of CRISPR and how it plays a role in breeding. And then to René Custers, who will look at the potential trends and applications of gene editing. We will then discuss with our panelists, Max Schumann and Irène tolleret. So Max Schumann is a farmer, and Irène tolleret is a European Parliament member. We will then have an interactive Q&A using Mentimeter, and we will also ask you some questions there. And then Mark Cornelissen will then make some closing remarks, and then we will close for the day. So for those of you that aren't familiar with Plants for the Future European Technology Platform, so short Plant ETP, we basically are a member-based platform. You can see all our members here, and they represent the plant sector and everything from fundamental research to crop production and distribution. And our main mission is to promote the flow of innovation to the market for better societal benefit. And by having this very mixed group of members, we try to share realistic views from multiple perspectives throughout the agricultural value chain. And we really believe that this is very crucial for proper decision making. Basically, in the context of the Green Deal that I'm sure most of you are already familiar with the EU going towards a carbon neutral society by 2050, there's a lot of goals and targets that are very ambitious, but it's also essential to consider, especially in the plant sector, that the time to market is not as short as some people might think. For example, we have the farm to fork strategy with goals for 2030, but actually, if you look at here the figure on the right, so on the left of this figure, you have the entire agricultural value chain, and then you have academia throughout. And basically, depending on where you will make changes, innovations, and research, the flow to market can be very different. It can be from anywhere from a number of years to all the way to 30 years. And actually, if you look at all the bottom for farm suppliers, these include breeders. And basically, developing new genetic material for plants is actually something that takes an extremely long time, and it can take from 15 to 30 years, depending on what the starting point is. So this is something that is really needs to be considered using traditional breeding methods. And more innovative breeding methods like gene editing can massively reduce that time. So basically, we are sitting on a huge wealth of barely exploited research data. I did a very quick search, for example, on PubMed, just with the words plant molecular biology. So looking at any publications, scientific publications that were published over time with these keywords. So of course, this is a very rough estimate, but you can see that in the past decade, there are multiple thousands and thousands of publications coming out. And really, genome editing is currently the most efficient tool we have to utilize this data for the greater benefit of society as a whole and to have more innovations coming to the market. So I think you're all aware of it, but I'll still touch on this because basically when gene editing was addressed at the European Court of Justice, basically it was determined that it should be regulated as a GMO, even though mutagenesis techniques, other mutagenesis techniques were exempted from this regulation. They are still considered GMOs, but they were exempted from the strict regulation. Even though gene editing is a targeted mutagenesis, it was considered that it should still be regulated as a GMO because it doesn't have this, this long record of safe use, which is a very vague term. And basically, according then to this ruling, then gene editing varieties, plant varieties, or any other organisms generated using gene editing would have to be regulated as GMO in the EU. And for some people, that is considered as being a ban on gene editing varieties. And although technically it's not a ban, if you do consider the hurdles that a GMO approval is, for example, the administrative and financial burdens to get a GMO plant variety accepted on the European market, for consideration, there has not been a GMO approved plant variety on the EU market for over a decade. And the time for risk assessment through EFSA is a matter of decades as well. So although technically this ruling is not a ban on gene edited varieties, it is as good as a ban because the restrictions and the burden is so high that it would be very difficult for companies, especially SMEs, to develop these products for the market. And also something that's important to consider is that currently 19 out of the 27 European member states have a full or partial ban of cultivating GMO on their soil, which means that basically even if some GMO varieties are approved for the market, they will not be grown in most of the EU countries. So basically, through our working group that we have at the Plant ETP on gene editing, we have assessed a little bit the impact of the CG ruling. And again, as I mentioned before, when you look at the time to market from discovery research to a plant variety on the market from anywhere from 15 to 30 years, we assessed that really the impact at the consumer level of this ruling will actually start to become visible in 10 years' time. And basically what's happening is once this ruling was decided, we realized that all the things before the farm, so all the farm suppliers, including academia, have started changing their tactics to adapt to this because they see that there's basically a competitive disadvantage that could occur. For example, we foresee that in the future, importing from outside of the EU might be more cost effective than local. There's also a problem with the critical scale of innovation. There's a risk of brain drain, career options that are reevaluated, study choices we have seen in the last few years, less and less people wanting to study plant biology because they don't see this as something that they can really exploit fully because of the restrictions, and also partly because of the mistrust towards new breeding techniques and things like gene editing. And there's also this cost of lost opportunity because then as time goes, and it has already started, Europe, the European Union, is starting to lag behind other countries like the US that has already started marketing their first gene editing variety. And if you look here on the right at this map, I won't go into the details, but it's really just to show the greener the color, the more welcoming these different countries are to gene editing on the legislative context. And you can see that Europe is really very red. We also have New Zealand that is restricting gene editing in certain ways, but it is really a stark contrast compared to the rest of the world in the direction that Europe is taking. So finally, also to put things a little bit in context, I mentioned the Green Deal and the farm to fork strategy. Things are looking up. We have, we have hope that things might be reevaluated, especially in the farm to fork. There is this quote here where new innovative techniques, including biotechnology and the development of bio-based products, may play a role in increasing sustainability, provided there are safer consumers in the environment while bringing benefits to society as a whole. And we definitely believe that gene editing and other innovative breeding methods could do that. And basically, for those of you that might not be aware, the European Commission is currently carrying out a study looking at the potential of what they call new genomic techniques, of which CRISPR is one of them, gene editing, I mean, and basically how that could play a role in improving sustainability along the food supply chain. So this study will be concluded in April of next year, and we really hope that this will open the avenue for maybe reassessing the legislation around gene editing. So just to finish up as well, there's also, I'm sure again that you've heard about this, the Nobel Prize in Chemistry of this year has been awarded to Emmanuel Charpentier and Jennifer Doudna for the development of the method of gene editing. And we're very happy about this because it really shows that gene editing can play a role in society for societal benefits. There, so this is my presentation. I will leave the floor now to Professor Holger Puchta, who will give a presentation about about CRISPR and how it can transform breeding. So Holger is the director of the Botanical Institute and the chair of the Plant Molecular Biology and Biochemistry at Karlsruher Institute of Technologie in Germany. So go ahead, Holger.
I try, wait a second. So you see, everybody sees my slides?
Yes, perfect.
Okay, thank you so much for inviting me, giving me the chance to tell you a little bit about basics of CRISPR-Cas and genome editing. As well as, sorry, as it was already mentioned, this year, the Nobel Prize was awarded for CRISPR-Cas, but the point indeed I want to make is that we have to go far more back to really understand more about the technology, also about the risks of this technology might have. And therefore, I have to first ask the question, the very basic question, what is breeding all about? And as you can see here, in the middle of this slide, you see a brassica plant. This is looking like a typical plant. Wonderful plant, and then you see all these different kinds of monsters, which some of you might really like to eat. Maybe they had them even for lunch today, like Brussels sprout or cabbage or broccoli. And the basic point is that there was this genetic potential to really, the breeders used to develop out of this plant all these different kinds of vegetables. But the problem, of course, and this is very typical for breeding in general, is that this genetic variability is lost in these crop plants. And if you just want to try to get to make out of a Brussels sprout kale, this will not work anymore. So there's a dramatic loss of genetic variability in classical breeding, and a very, very brutal way to change this was already developed long ago. And in principle, this was the first time when really double-strand break induction was used for breeding. And the way this was done, and this can also be done with chemicals, I concentrate now on radiation. You just take the plant, put it under a radiation source. This is also shown in this picture here. This is a cobalt source. These are all the plants around it. And then these plants will be massively hurt, and the DNA will be massively hurt, and genetic information will be broken. And also, this is a brutal way. In principle, what you get in the end is genetic variability. So you see here a break is introduced in the DNA, it has to be repaired, and something new arises afterwards. So in principle, you might argue, well, but this is only killing genes, how can this ever lead to something productive? But you all, of course, have to see, if you think about breeding, if you think, for example, about grasses, in nature for grasses, it's very important that they grow very fast and have very long stems. But for farming, very long stems are very bad, because if a storm comes, the storm will kill all the cereals, and what you need is short-stemmed cereals. And so if you knock out the gene for growing long stems, you will get short-stem cereals. And this is why there are a lot of traits that are interesting for breeding that can be achieved with this technology. And in principle, you have to see that this technology has been used for 70 years by now. I don't know whether you had a grapefruit today, but most probably this grapefruit was seedless. And all seedless grapefruits like Ruby Red were also developed by this technology. If you had a beer today for lunch, or maybe yesterday in the evening for dinner, all short stem barley, in principle, is also developed by this technology. So worldwide, we have more than 3,000 cultivars that were grown for 70 years, and they were all produced by double-strand break induction. The problem in all these cases is, of course, that besides the break that is in principle giving us a good mutation, we have a lot of other mutations introduced, other breaks introduced that also have to be repaired. And so we have a lot of so-called offsite effects. I continue, sorry. In principle, for a long time, it became clear that this is a brutal method, and nature does it, of course, better. So why don't we go bio with this kind of technology? And in principle, to go bio means to see what nature does. How does nature do it? And during the lifetime of all organisms, you have this problem that you have to make new combinations of DNA. This happens, for example, also during your lifetime if you want to produce antibodies, or if you inherit your genes to your son or to your daughter. You have chromosomes, and these chromosomes, it carries the genetic information, but these chromosomes, you don't inherit it in a piece from your father to your son, for example. There's a lot of exchange going on, and for this exchange, you have to cut DNA and you have to re-ligate DNA. And this is also, as you can see here, occurring during meiosis. And this is a very general phenomenon in all organisms. So molecular scissors are used during the lifetime of all organisms very regularly. So this is nothing new, nothing specific. This happens all the time, everywhere. But of course, for us at this time, it was a challenge to, in principle, use then molecular scissors for inducing directed breaks. And in the first step, we were very happy, and this was already more than 25 years ago. In 1993, we applied, and I was very happy that I had the chance to work at this time at the Friedrich Institut in Basel, and I was really the first, in principle, to really test what can be achieved if we induce a break. And we used this time having these enzymes. In the beginning, we have enzymes that only were able to cut at one specific site, but later on, people developed enzymes where one could really address different sites in the genome. This was done in the beginning 2000s mainly. And during this time, we not only learned what we can achieve if we introduce a break, but also risks, also other changes that might happen with this. So in principle, the take-home message of this slide is that we have a long time experience with all these technologies inducing double-strand breaks. And this is nothing that just was invented in 2012. The problem, let me state this, the problem with all these elder molecular scissors was that they were not very efficient and they were quite expensive. So you had to wait, for example, for a year to get such an enzyme, and you had to pay 25,000 euro. So everything dramatically changed, of course, when the CRISPR-Cas technology became available, because here, in contrast to all other enzymes, you have a small RNA molecule that you can easily exchange, which is defining the site of the break. And this makes this system so much easier and so much cheaper. So now you will spend 25 euros in principle to construct via DNA such a new site, and you can do it in a day. And this is why the revolution took place with CRISPR-Cas. And this is why we see this explosion in biology that compares with nothing else since 25 years. And so in principle, if we think now about it, what can we achieve in plants? We can really achieve a lot. So CRISPR-Cas is more efficient than anything else before to induce specific mutations. We have in principle almost no off-site mutations. So these scissors are very, very efficient, just targeting this one and only site. And now we can target with this flexible RNA system any specific sequence anywhere in the genome. And René will tell you in a minute for what kind of fantastic approaches this has already been used to improve traits in crop plants. I don't want to go in too much detail, but I want to make a very important point now. And this point is directly related to what Amrit told you before about the EU ruling. So in principle, think about what is regarded as safe by the European Court and what you find in all fields everywhere in Europe. These are, for example, if you go to a barley plant, these are plants that are produced by classical mutagenesis. And if you look at the numbers, you will find 10,000 mutations in comparison to other barley plants that are induced by this old kind of mutation induction. And then, and I don't think that anybody or most of you are aware of this, if you just take two natural plants out of a barley field, are they similar? Do they have the same sequence? No, even these two plants differ by 100 nucleotides. This is the natural mutation rate. And if you compare this now to what we can achieve with CRISPR-Cas, making just one specific mutation, you realize how far we are gone and how safe this technology is. And even if you might argue that there are two or one off-sites, this is still beyond the level because it's beyond the natural mutation rate. So this technology is really so if it comes to safety. And now I'm not able to, yes, I can. So just to sum this up, scientific risk assessment, double-strand break repair is a natural process. Mutations occur all the time. So if we induce by molecular scissor a mutation, you cannot discriminate it from natural mutations. Plants with CRISPR-Cas induced or spontaneous mutations therefore cannot be discriminated. And therefore we can really and truly say CRISPR-Cas induced plants are nature identical. Classically mutagenized plants are extensified due to the long safety record by the court. But if you see the scientific data, you have to say edited plants are at least as safe as mutagenized plants. And I tell you, I'm convinced they are more safe. Okay, so far to the current state of art. I was asked also to look a little bit in the future. What are the next steps? What can we expect from this technology? And we can expect much more from this technology because this technology enables us by using different RNA molecules to induce more than one break at a time. And this is really a major advantage. So what is breeding stuck with at the moment? So we have a lot of wild species related or also not so related to cultivated species that have a lot of interesting traits, for example, heat resistance, salt resistance, pathogen resistance, and it's very difficult to cross them into cultivated plants also because in crossing you cannot just simply combine genes. Genes are organized on chromosomes and part of the genes are on parts of the chromosomes where no genetic exchange can take place during the breeding. And if genes are closely located on chromosomes, you cannot exchange it. And so there are two modern strategies that have been developed based on CRISPR-Cas to overcome these problems in different ways. One is to induce several mutations at the same time in a wild plant and thus make a wild plant transform it to a cultivated plant. So you have to see, and also this was mentioned before, that there's a lot of research has been done on molecular biology of plants and all the different developmental steps. So we really know by now how wild plants differ by cultivated plants. They differ by a few mutations that make fruits better, that make fruits more well-tasting, and in principle also make more fruits on this plant. And if we change these genes by addressing them with CRISPR-Cas, we can really really make a wild tomato, as it was shown here and done by Dan Woiter's group 2 years ago, we can really transform a wild tomato to a domesticated tomato. And I think this is a very important development for the future, because it gives us the possibility to also take plants that are very well adapted to heat stress or to soil stress, if you think about the global warming problem, or to different resistances to reduce pesticides, and transform these plants to cultivated plants, like it's shown here in this picture for ground cherry. But this is not necessarily restricted to ground cherries. This can be done with several plants that you might not even be aware of, but in future might be new kinds of crop plants we can develop. So this is really a very exciting development. And I also just want to finish this story with a recent development in our own group. The problem with breaking genetic linkages, which I also told you before. So the problem is that I have two genes. For example, I have a gene, this red gene that gives a taste to a tomato, and on the other side you have a gene that in principle is responsible that a pathogen can attack this gene. So you want to get rid of the one gene but not of the other gene. If they are on the same chromosome, you cannot do this. But now we developed a new kind of technology, not only editing genes but also editing chromosomes. Chromosomes change in nature also all the time, so this is nothing really strange thing that we are doing. We are just exchanging here chromosome arms, and you see here now this gene is on this chromosome, this gene is on the other chromosome, and then you can segregate these two traits away from each other. And I don't know whether you see this. I don't see it on my computer. Here on this side you might also see a molecular structure of the chromosomes. You see on the upper part red and green dots, and in the lower part these are new combined. And so this is the first time that we really could achieve to get chimeric chromosomes in plants by using the CRISPR-Cas system. And with this, I want to finish my presentation, not without thanking you for your attention, but also, and this is for me very important, to also thank the European Commission for funding us over all these years so efficiently. I was very happy to get 2 times a ERC advance grant. Many thanks to the European Research Council for this. This really enabled us over all the years to keep pace with the other groups on the other parts of the world, not to lose contact to the research community in gene editing. And with this, thank you so much for giving me the opportunity to tell you a little bit about the most recent developments that I think are really exciting.
Thank you very much, Holger, for this really interesting talk. And I think this has given a lot of ideas to people, maybe to some people that weren't so familiar with the technology. So I would just ask if there are any questions just for clarification. We won't address the main questions and get into the discussion at the moment. So I'll just ask Nick, who's moderating the questions.
There are no particular questions for clarification at the moment.
Okay, then before we move on to the next talk, we'll look at our poll about what type of stakeholders we have present today, just to get an idea a bit of the distribution. Okay. Great. So I see there's quite a big representation of academia and public research. Also a big percentage of people from breeding and seed sector. So I think these we can consider are already quite familiar. But I'm very happy to see there's a very good representation of farmers, as well as public authorities, even people who are representing themselves and are interested, members from civil society and others, and also retail and processing. So I think there's a little bit of everything, and we're very happy to see that. So welcome, everyone. I see about half of you have answered that, so that's nice to see. We also have a follow-up question to the audience. If you could share your screen again, Nick. There. So again, if you're on menti.com and you enter the code here above, we'd like to know from you, your personal experience, how you feel the ECJ ruling regulating genome editing as GMO is affecting your work. So this is just a word cloud. You can just enter 1 or 2 words, and we'll just have a look at how that might be growing. So please go ahead. Okay, that's a nice start. Oh, okay. Well, it's nice to see lots of things coming up. So I definitely see a lot of negative things associated to this, or at least the impact of the ruling. I'll just give you another 10 seconds or so to keep filling up, but it's growing very fast. Okay, so it's stabilizing a little bit, but I definitely see things like frustration, restrictions, stopping innovation. There are quite a few people who are not affected, as we can see here, which is still quite big. But yeah, there is a lot of mention of restrictions, challenges, difficulties, and of course that would vary depending on what your field is. But that's very nice to see. Thank you for that. So you can keep filling that out if you'd like. It would still be available. Meanwhile, I will leave the floor to René Custers. So René Custers is the director, or I'm sorry, the regulator and responsible research manager at VIB here in Belgium, and also at EU-SAGE. So EU-SAGE is an academic organization for European sustainable agriculture through genome editing. So go ahead, René. The floor is yours.
Yeah, thank you, Amrit, for introducing me. I'm going to present, well, and thank you for the opportunity to give a presentation here in this webinar. It's an important topic, I think. We already had a very nice introduction by Holger on the more technical aspects and the prospects from a technical point of view that genome editing proposes. What I would like to begin with is a little bit the broader picture that I would like to present and show some context for the discussion on genome editing in food and agriculture. I think we all know that there are very important challenges that we have today in terms of food security, in terms of climate crisis. It is very important today to work on biodiversity conservation. Species are going distinct. Lots of things are happening in important forests worldwide. Our agriculture and food system is still very dependent on very heavy inputs. And all these things, I think, will affect the sustainability of the system and the sustainability of the agriculture and food production. I think in the situation that we are in today, what we experience also with the COVID crisis worldwide today, I think shows, well, is an excerpt of how we as humans put an awful lot of pressure on this earth. We are feeling that more and more with the climate crisis, with the pandemics. Of course, pandemics have been there before in our history, but I think in the current situation with the growing population, this is something that is growing, will be becoming more, well, will be not that rare anymore. It is something that we have to deal with. So within agriculture, I think there are these very important main challenges. And what we're looking at today and discussing today is especially from the angle of crops and genome editing, genome edited crops. And I think we can safely say that well, improving the genetics of crops, introducing properties that have been improved, that have certain result in certain traits, of course, can contribute to sustainability. And sustainability, Amrit, you already mentioned that in your own introduction, referring to the European Green Deal, but also, of course, on the global level, similar challenges have been formulated under the UN, and I think these are this is the big picture that we should be looking at. This is something that you also already mentioned in your introduction, even showing a graph with numbers of publications in past and recent years. Within the audience that we have today, there's a lot of people from academia. We are all witnessing the acceleration in genetic knowledge that we have witnessed over the past years. Learning about what specific mutations are doing in crops, which mutations affect how crops interact with their environment. So there is lots of knowledge that is accumulating. That knowledge is being exploited in all kinds of different ways within the breeding sector. But of course, as you all already mentioned also yourselves, I think the tool of genome editing, CRISPR and other technologies is by far the most efficient way to exploit that knowledge. Knowing which mutation has results in which trait, those are things that we can much more easily introduce using these technologies. One thing I would also like to point to, everybody's talking about genome editing today, but genome editing, of course, is not something that, there have been many more innovations in plant breeding before, from crossbreeding, hybrid breeding, cell cultures that have been introduced, mutation breeding, genetic engineering, recombinant DNA technology in the beginning of the 80s. And of course, today, genome editing is added to that toolbox. And the crops that we have today and the crops that we will be introducing in the future will be an accumulation of all that different technology. It is not that genome editing will replace all these things. It is something that is going to be added. In certain cases, it will be used. In other cases, it will not be used. But I think already today we are in a situation that if you would label in the supermarket the different foods that we have there, in terms of what breeding technology has already been used in there, well, genome editing is just one additional thing that is going to be added on top there. There are different ways you can look at what is already happening in crops and which types of things are being introduced using genome editing. Research colleagues from the Julius Kühn Institute in Germany have already looked at literature and screening literature and making reviews on the different things that have been appearing in the scientific literature. On what types of crops are being edited and what types of traits are being introduced using editing. Here I'm presenting one other approach to look at this, and this is just a simple excerpt from, I took a simple look at the USDA-APHIS MI regulated database. That's a database that started in 2011, so almost 10 years old, where actors can ask the question, well, I have introduced this modification into my organism. Does that trigger the legislation in the US, the USDA legislation, yes or no? And you see that has been, in the beginning, slowly growing, but I was amazed to see that in 2020, this year, and the year has not ended yet, there were already more than 80 examples, 80 cases in that database. These are not all plants. These are also not all CRISPR plants, but the majority of those 86 is related to CRISPR technology and crops. So I think this database shows like the way scientific literature is showing that in reality, I would say genome editing in crops is really gaining momentum, and the examples are coming out there. I think this graph is a little bit more important than looking at scientific literature as a whole and taking information from there in looking at what types of crops and what types of traits have been introduced. Because while scientific literature, there's an awful lot of there that is just being done in research and for research purposes. While I think here in the MI regulated database, you will see much more, well, it's much more limited, I think, to those crops and those traits where people have a real interest in actually moving those things forward to the market. So I would invite everybody to have a look at this database and see what the different examples that are presented in that database. And I will have a few pictures of things that are popping up. There are many more examples than just these. So high fiber wheat is an example. There are as an example of drought tolerant maize. We have mildew resistant wheat that has been produced already with genome editing. And here from the MI regulated database, you can also find different varieties of potato that have been altered using genome editing technology. If you look at the properties that have been that are being introduced, we can all relate them to the first picture I showed to you in terms of food security, climate change, inputs, and also biodiversity conservation. Because if you look at the crops and if you look at the properties, you will appreciate that a drought-tolerant maize is something that is beneficial in conditions where climate is changing, temperatures are rising, and drought is becoming a bigger problem. Mildew-resistant wheat is very important in the sense that this is an example of a disease-resistant crop variety that will help us lower the use of pesticides. And that's a very important goal also within Europe, within the Green Deal. And I think these examples are a very good illustration of how certain things can help contribute to sustainability. Another set of examples here, and the strawberries, I found an interesting one where an edit has been introduced that allows additional flowering. So you have a crop that will be able to produce more fruit over the year. The grapevine is something that is I also find a very interesting example, because fruit and grapevine and fruit trees are crops where you don't have rotation, and where you are performing agriculture where you don't have rotation. That's where you get big problems of certain diseases popping up in those in those crops. And within grapevine, fungal diseases are a big pest, and fungal resistance is something that is very beneficial there and can be introduced using genome editing. The advantage of genome editing here is also that introducing a small edit resulting in a fungal resistance will leave the Chardonnay grape still being a Chardonnay grape. So it is a perfect blend of modern technology with tradition. And that is something I think that is also very important in the context of genome editing. Other examples here, but again, I would like to invite everybody in the audience to have a look at the database and see what things are coming up. Also, new things will be added in the months and years to come. And this is really showing the potential, and well, more than the potential, I would say. Looking at these things, I think I see a difference with the period that we had 25 or 30 years ago, or already almost 40 years ago, when we started recombinant DNA technology was introduced. where genetic modification was also thought to bring all kinds of benefits and be at the basis of an awful lot of new developments within crops. I think in reality, genetic modification in crops using transgenic technology has delivered a number of crops, but in practice, the amount of traits that have been introduced using a traditional genetic modification is not that wide. And what I see here, I think that in genome editing, the promise that is there with the technology is also being delivered much more, I think. Because I think we see the amount and the diversity of beneficial properties and traits that are being introduced using genome editing is really growing. So in this short period of time, and the diversity of traits that you see popping up in all kinds of different crops is showing that the potential is starting to really become just more than a promise. I think also what is important is the speed at which this is being introduced. I see things more rapidly moving than with the transgenic technology that was. It is also true, of course, that today we have much more genetic knowledge and knowledge about what the effects of mutations are than we had 25 or more years ago. Not all traits will, of course, improve the sustainability of a crop. Quite a number of traits may be neutral in that sense. So it's not that every genome edited crop will be a contribution to improve sustainability. I think we have to be realistic in that. But I think that will be, well, policies like the Green Deal, the problems that farmers have, I think those will be directing breeders to make choices in which types of edits they want to introduce, and I think they will also look at these pictures and try to do their best. We're talking about the crops and improved genetics as a contribution to more sustainable agriculture and food production. Of course, we also have to be frank here and say that is only one little piece of all the different things you can do to improve the sustainability of agriculture and food production. I've listed up a number of additional things here. There is more than just this here, but I think it is important to state that, a breeding toolbox is one thing, improving genetics is one thing. It is not an either-or thing that we should do. I think it is important that we combine all these different approaches and each have their contribution to a more sustainable food production. Looking at the future, I think in terms of the potential of making crops more, contributing to sustainability, I think with genome editing, we will be able to realize those goals in a faster and in a better way. And I think it is seeing what is happening internationally, I think it is not a good thing to block genome editing. I think that is not a very responsible thing to do. But of course, then the main question for all of us, and I think that is going to be, one of the major things that we can discuss further in this meeting is how are we actually in Europe, and that is the context that we're discussing, how are we going to allow genome editing in a responsible way. And with that, I would like to thank you all for listening, and once more, thank you for giving me the opportunity to say some words here.
Thank you very much for that, René. That was very interesting. I think it really sets the scene. So before we move on to our panelists, Nick, are there any clarification questions either for René or from Holger's presentation before?
Yes, there is one question for Holger, and I think that's related to his mentioning of the EU funding. The question is, Why the EU funds research in this area if it will not be used to develop EU-specific resilient agriculture?
Well, all I can say is that all my funding, all the decisions for funding me were taken before the EU ruling. So at the moment, as far as I know, there is not so much written out where gene editing technology is involved to improve crops. But I only can say this, of course, for the national programs in Germany, where we really are in a very difficult situation as basic and applied scientists, because we really have tremendous problems to get funding. Also, I think we are doing really interesting and nice things. So that's a little bit, or it's very frustrating, to be honest.
Okay, thank you, Holger. There is another question for René. It's related to the term sustainability. More concrete, which traits would you regard as neutral in view of sustainability? And is it environmental sustainability only?
One example of a trait that I would regard as neutral is a change of color of a fruit, or a change of color of the vegetable, things like that. I think you can think of all kinds of traits that are somehow related to, well, quality related stuff, but don't immediately have an effect on reducing inputs, lowering carbon emissions, etc. When you talk about health related traits, improving the healthy properties of foods, that can also, of course, also be regarded as a positive trait and contributing to sustainability.
Okay, thank you, René. There is a small follow-up question on that. In your view, how should the sustainability aspect be accounted for in legislation for authorizations of genome-edited products?
Well, the question is whether or not sustainability should be a criterion for before allowing something onto the market. I think in that respect, we have to treat crops that we cultivate in the same way as we treat other products that we introduce onto the market. I think it would not be fair to introduce sustainability as a criterion only for instance, for genome edited crops, where we don't apply a criterion like that when we introduce, for instance, a new food additive or something else. Right?
Okay, and there is one clarification question. It's about the database you were referring to. Where can we access the database of crops that you mentioned?
The MR regulated database is simply, if you Google with USDA APHIS and MR regulated, you will immediately find it.
Perhaps if you don't mind, you could put it in the chat, then people would have the link. That would be great. Thank you very much for that. And thank you, Nick, for the questions. I think, so I see some people are asking questions in the chat. Please go to menti.com. I think there are instructions in the chat where to go and what code to use, and ask your questions there. For sake of time, we will address and have a longer Q&A later where we can discuss with all the speakers and the panelists. But right now, I'd like to move on to our panelists. So we will be starting with Irène tolleret. So she is a member of the European Parliament representing France, and she's also a wine grower, so also representing farmers. And basically, she's a part of Renew Europe. So thank you, Irène, for being here with us. And we'd like to hear, your thoughts about gene editing and what role it could play in sustainable agriculture.
Yes, thank you very much for having me here today. It's very, very interesting. We have got a tough one in front of us, because with the development of technologies and knowledge, we can, and computers, we can have more and more and more very small information and enrich our beforehand knowledge. And so we may be scared about innovations. But if you look at it, innovations have always arrived, and they have always scared. And some of our traditions today are innovations that are not so old. In the wine trade, stainless steel did not exist, all the coal did not exist, frozen food did not exist, a lot of things, climatization, air conditioner. So our life is with a lot of innovation, but at the moment, we are in the middle of a pandemic, and we are talking about, with genetic edition, we are talking about things that really scare us, because it's a Frankenstein sort of feeling. But at the same time, we have a pandemic. The scientific talk is not respected anymore. I'm sorry to say that, but we have had on the TV numerous doctors talking about COVID and wearing masks, and we have had strikes saying no, they don't know. And so we have at the same time a double crisis of masses of knowledge and through media communication, very simplified and fake news sort of information. So for members of European Parliament, how can we deal with that? So first of all, I think that we have only one planet, and to meet the challenge of the climate change, we need to improve our sustainability, and we won't do it without changing things. So we absolutely need to develop highly resilient cultures. We can do it the old way, as was shown since 1,000 years ago. It will take time. Or we can put on the table that there are new techniques like CRISP arriving, and what do we make of them in a legislative way. It's also important that we reason a planet compared to Europe. Because European farmers, if they have to complain with the ambitious objectives that are proposed by the European Commission to reduce pesticides and fertilizers, but at the same time, they have a lot of impact of growing extreme natural phenomena due to climate change. The drought that has been talked about is affecting the south of Europe, but it's coming to the north. And we have also the risk of pandemic and epidemics that we see, and we have a lot of new illnesses coming. So how do we tackle that? I think it's so very important that we develop selection techniques to improve plant resilience. But what I was saying, Europe is part of the planet. And so we really must not have Europe, as it is today, lagging behind compared to other countries like the United States, Canada, Brazil, Argentina, Australia, New Zealand. All these countries take a positive view to new breeding techniques, especially those that do not involve the manipulation of foreign DNA, as is the case of CRISP. And what is the risk? The risk is that we end up in the European Union preventing the development of new breeding methods, but at the same time allowing import from these techniques from third countries. So we really need to put things on the table and to confront them to sustainability goals. Genome editing, for me, should be compatible for organic farming and other agroecological systems. Of course, we need the acceptance of these techniques on several factors. First, any new technique must be validated by scientific authorities to avoid any possible suspicion of dangerousness. Second, public opinion must be must be fully informed of the benefits of the new techniques. We must talk about the benefits, not only of the risk, to curb emotional reaction without scientific basis. These techniques must protect or even improve biodiversity in the European Union, and we also need a very clear legal framework to be able to develop these techniques while avoiding any ambiguity or legal and illegal uncertainty. I think the ball is in our hands, and we need to do a perfect match with it. It's an opportunity, but it's a big challenge of collective intelligence. Thank you.
Thank you very much for that. It's really nice to have your input. So just maybe a follow-up question, because wondering, so you mentioned there's a lot of, we're getting more and more information basically, and very precise information, but a lot of it is also fake news. And I think a lot with the internet now, in the end, people will get the information that they're looking for. And I was wondering, so I know myself in the past as an academic, I have also done a lot of outreach, and I know many people that have done it. And we're always told also from the Commission, scientists need to communicate more. But we are trying, but maybe you have some tips on how we could better communicate or better address some fears.
I think that what you are doing today is already a dialogue. And also, sorry, I will be very blunt. Sometimes a scientific, when they talk, it's completely boring. So I think you should add a sexiness to the way you promote your techniques, and especially by addressing the objectives and the results. At the moment, we are going to change the legislation on pesticides, but I can tell you as a member of the European Parliament, each time we talk about a pesticide, we never even talk about the importance that this pesticide has on any sort of culture. So that means that we need to relate like there is the climate change. With the climate change, the drought is coming very big in these places. That means World Health Organization and UNO have official communications on that we risk so many 100 million people more dying of hunger if we do not tackle the subject of drought. So now that the subject is on the table, how do we solve the subject of drought? We can try to put water, but we can also maybe develop plants that are resistant to drought. We can do that in the old method or in the new methods. And then, yeah, so not talk too much about the very important things that for the scientists make the breakthrough, but maybe talk more about how these techniques will change the life of citizens that do not care a jot about Christ, but want to eat.
Thank you. I think that's a very good advice. So we will now move on to Max Schumann, our other panelist. So Max Schumann is an arable farmer in Finland. He's also representing the Finnish farmers organization MTK. So welcome, Max.
Thank you, Amrit. I hope you can hear me well. It's very nice to be here, and it has been a very, very interesting already discussion going on. I will now bring you in a little bit into the farming sector, into the discussion as well. And I was thinking about starting to set just the scene under what we as arable sector, not only in Finland, but all over Europe actually work. Firstly, the first thing most of the farmers would do, you wake up in the morning and you would look what kind of a weather it is. So the climate around us plays always a very important role. Since the arable sector, we are growing under the so-called big roof. That's why the weather has always been there. And we all know that the climate is changing around us. It has always been changing, but now it just has picked up a little bit the pace in how it changed. And it is much, much less predictable, at least as it feels now. Then we have the market that has always been there. We have always had to look at the market. For us, I mean, in Europe, what is needed, what crop should I grow, where is the market going in related to the price. And then we also have to look at the world market, since the arable sector is very, very much dependent on what's happening around us, and we are very open. So also the world market will play in there quite a big role, meaning that we are right away into trade, exports, imports, etc. So we have there a competition ongoing in between the crops, but also the markets. And then we have a new one that has been here already for a while, but it feels that it's getting to be a more difficult one to tackle, and that's legislation. We get more and more legislative things that we really have to take care of. And for sure, some of them are related to the market, but more and more today are related straight to the climate. So what we have been talking here about sustainability, you have the sustainability that I believe that has been discussed mainly here is the environmental sustainability. But as a farmer, I'm also looking at the economical sustainability, because I also need to survive in this kind of a world, in the competition with the outside European market, but also internal market as well, in between the different regions, as it is already today. And then for sure, I have to compete with the climate. So for me, the plant itself is the most important thing. I grow crops on my fields. I need to make sure that these crops will perform well, produce to me quantity, but also quality. It's not only the quantity that is the key thing that is usually looked at when you look at the market, it's also the quality. And that doesn't mean that it is always just the cereals we are talking about, but because I'm an arable farmer, I like to have a good crop rotation. These crops are on yearly basis. We are not talking about perennial crops, but we are talking about yearly basis. So I need some good plants into my rotation. And these crops should also fit in then to the market. This is how it was before. Now I have to fit the crops also in to fulfill the legislational needs as it is. So for instance, what I would do, I would bring in, and I have done it for many, many years already, I have kept in also either oil seeds or then nowadays more and more protein crops. And here I see that the European Union, I mean, we are lagging behind. We are not growing enough plant proteins. And I can see that in my own country we need more of it, but I know that on EU basis we need more. And that is also what now today the society is starting to demand, and also legislation is trying to push this way. But for me as a farmer to start to grow these crops, I need to make sure that they can compete firstly with the crops I already can grow today, meaning the cereals that we have already been able to make very high yielding, very, very good quality. So I have to make sure that it is, for me, economically sustainable to bring in these protein crops to my plant rotation so that they can also produce plant proteins, yes, but also at the same time, they need also to take care of my economical sustainability. I also need them to be able to compete and bring in something at the same level as the cereals, or hopefully a little bit more. So these are the things that I'm looking at. And here I know, and here I see already, I've been a farmer since 1986, quite a few years. And here I can see that at least the plant breeding has done me good, a lot of good. Different types of plant breeding systems, etc. They have always been able to perform, bring better yielding, better quality into the crops that I grow. And this is, I hope that this will also continue. Now I just see that there is a lot of stress coming from the very, very fast changing climate. So there has come more pests up here into the north, but I also know all over Europe, it's not one size fits all. It hits everybody. We also see that there are more new types of diseases coming in as well. So we need crops that can be stronger and more resilient to both disease and also to pests. At the same time, as I would like to see that they would still perform the same and even better. And if I would at the same time be able to reduce inputs, fertilizers, and for sure plant protection, I would be very happy, because everything that I can save here is a cost saving, but it is also saving on my time at the same time. So I would really like to see that we would be able to use better the plant breeding possibilities we have today by bringing in new plants that can really help the farmers. Through that, it will also help the society. We would get our plant proteins locally grown. We would also be able to make sure that the social aspect of the sustainability would be at some point at least taken care of. We would have living countryside. We would have there most probably some smaller enterprises coming up, etc. So we could see that these kind of a thing, just talking about plant breeding and new varieties, maybe new type of plants coming into our rotation, can actually fulfill all three of the sustainability criteria that I put forward. And this is just plant breeding. And I think that these are things that at least me as a farmer, I would really like to make sure that we have the possibility to use the technology, to have a big toolbox in the hands of our plant breeders, and through that also a big toolbox in the hands of us farmers that will provide food, feed, fuels, etc. into the European Union. But also, as mentioned by Irène, we see that there are more and more people worldwide also needing more food. Europe is a very important place where we can grow more than what we can use here. We are big in exporting both cereals but also food products, high quality food products even. That brings for sure more euros back to European Union, but at the same time it also makes sure that elsewhere in the world also will have more food available. And at the same time our legislation is very strong, so we can say that they are very sustainably grown. So I think that this kind of a win-win-win situation could be created by bringing in new and better varieties. At least me as a farmer, very simply thinking, I would like to make sure that I can produce more on the hectare that is my production unit. I would really like to see that. I would like to be able to choose and change what I'm growing there according to the market needs, according to what the consumers would like to have, but still maintaining and taking in consideration that I am also economically sustainable towards my own family, my farm, and in that way also create certain jobs and possibilities for the social society around me. So I think that we need to make sure that at least the new breeding techniques that we know of today, at least will have a chance to show what they can do. Doesn't mean that everything will be done through them, but at least we would be able to see what can be achieved. Thank you.
Thank you very much for that, Max. It's very inspirational. I maybe have just a very short question. So you mentioned a little bit the economics. So maybe how would you foresee things would happen for farmers within the EU if methods like gene editing remain regulated as GMO? What do you see the impact would be for your competitiveness, for example?
I would say that it maybe in the short run, you would not be, how would I say, such an extremely hard hit, but already in a medium run, it could be quite devastating. Could be that we cannot keep up anymore with the production amounts, with the level of production, but I'm quite sure that we will see through the legislational push where you have to use most probably quite a bit less pesticides, PPPs in a whole, plant protection products as a whole, but also if we have to start reducing a lot fertilizers and bring these in, so we will lose in two ways. Firstly, we will lose in the quantity, but then when we start to lose in quality, it will start to hit also our industry. So the food industry will also have their hit right away, and this will then start to hit much bigger part of the European economics as well. So I see that already in a medium term, it might have quite heavy consequences, not only to the farming sector, but to the whole food chain. And I don't think that this is something that the European consumer or citizen would like to have. I think that they would like to see that we would have high quality, safe and nutritious food in the shops, on their plates, also in the future, and that it would be of European origin. We have very good foods here, and we have a very good food culture. Let's try to keep it.
Thank you. Yeah, I cannot agree more. So I think we will move to the Q&A so we can get everybody together, including the speakers and the panelists. But before we start addressing the questions, I'm sure there's a number of them already. We will just open for a few questions for you. So Nick, if you could share your screen for the mentimeter question. So again, we have just two word clouds, and we'd like to get some idea of how you see gene editing as regard to the legislation. And just a second. Here we go. Yes. So again, just some word cloud. Basically, what main challenge do you see besides legislation for plant genome editing to contribute to sustainable agriculture? So we have consumer acceptance. That's already coming up there. Yeah, public acceptance, public perception. Yes, I'm not surprised to see public perception and consumer acceptance being some of the top ones. Yeah, so I think we all agree in the fact that besides legislation itself, it's about perception, acceptance, especially from the consumer level, politics, of course, and how they can enable more acceptance. And that comes down to what Irène was saying, that we need to communicate better. And I think that's something that cannot be said enough. We need to find better ways, more relatable ways to communicate more and more. So a follow-up question, and we had kind of anticipated the main results of this. Basically, what do you see as an appealing plant genome editing success story that could convince society that genome editing could play an important role here? Any thoughts?
This is a question to all of us, or?
To everyone, yes.
A thing that in my discussions always was brought forward to convince the public, if the development is far enough, was, for example, gluten-free wheat, because this immediately shows benefits to consumers.
That's true. I think having something, again, that's relatable, that's applied to an issue that consumers might have, that could be more relatable. So I see here, it's a lot about traits. So drought tolerance that René was touching on as well, less pesticide that Max also mentioned, and that's part of the farm to fork. By 2030, there needs to be a reduction of 50% of pesticide use. So that's definitely something. Hypoallergenic, so as Holger was mentioning, allergies. Yes, so I think, these are very related to some of the things that have been put forward in the farm to fork. And I think this is, as Irène mentioned, we need to focus more on what we can do with this technology and not so much on the technology itself. So thank you everyone for that. So we will now move on to the Q&A. So Nick will then present your questions. So we will, as I said, take them in order of the votes that they've received. So please, if you have any questions you want to get answered, just vote on them on the thumbs up. Go ahead, Nick.
Yes, there is a first question for Irène. It is like, how do you see the development around the German Green Party opening up for new technologies such as genome editing? And in relation to that, what is the perspective of your party towards this?
Alors, I will give you my frank testimony, but I'm a new member of the European Parliament. So I would say that if we look, Renew is really the group, the progressive group. So we tend to want to liberalize everything. So that means that we would be the group that most probably would be the more pushy to authorize new breeding techniques, maybe with the EPP, in the sense that the EPP is always fighting for the freedom of companies to develop things. However, when we talk about the Greens and some members of the Socialists, but also the GUE, then we have like, oh, Irène, you are funded by Bayer Monsanto, you are a horrible NEP, and you want us to eat GMOs. And so this is why I really insist on the fact that we need to come through the other way. Do we want to have less pesticides? Okay, how do we do it? And as soon as we start saying, how do we do it? Then there is the advocates of the Middle Ages that will say, we do like we did in the Middle Ages, and so we will use very bad products, because in the medieval ages we were using asbestos, horrible poisonous natural things for our crops. So lead, bad things. So it's important that we arrive with the objective 50% less pesticide, and that through all the groups we explain to them that some things are just not possible, and that it's not possible that we kill a legislation just on hypothetical things that could be done. If they want to experiment around Berlin on how you have discussions with MEPs that explain to you that the agricultural model is Cuba. I mean, what has Cuba brought to agriculture worldwide? So we need to have facts and also to have experimentation protocols. So between the research and the consumer, there is the industrialization of experimentation. And I think that we lack a little bit on that part.
Thank you, Irene. There is another question for Max Schulman. Mr. Schulman, what would be your response to citizens that ask for no use of genetics in the solutions?
That's a very, very good question. I think there is one thing that was already mentioned by René. I think that what we need to do is just to make sure that the citizens will be informed. When they buy their products, you can today actually very easy have their labelings. I mean, they can fit quite a lot of information today on QR codes or barcodes. People could check and see. The same goes for the farmers that would buy the seeds. If you should also know what and how this new variety has been done, what kind of a breeding technique has been used, so that we have, after a while, actually will be able to pass on the information through our nowadays digital world, so that we have a good traceability, so that the farmers but also the consumers would have a choice of themselves, of their own, what to take in, what to buy and what to eat. And another thing, just to back to the question before, I think it's very, very important that we can wake the interest of different groups, different people groups, political groups, to learn more and to want to learn more about these tools, what can be achieved by them. That's the only way how we will be able to get this discussion to such a level that we actually can have something use out of it. If one part just says no and the other one says yes, we are in this yes-no dilemma. Thank you.
Thank you, Max. I see René raised his finger. If you want to comment on that.
If you don't want genetics in crop, then it's going to be very difficult, because genetics has always been there, and the things that we have been doing from 10,000 years ago until now is all based on genetics. But compared to what we are doing now, in the old days where we were doing blind genetics, while now we're doing much more targeted genetics and know much better what we do.
Thank you. Is there anybody that would like to add on to that or to what Irène was saying?
I think, Amrit, there's a more a general question for everyone. It's a very pertinent one. It's like, who is responsible to get the public aware about this scientific innovation? Because we have many different societal actors, but who is in the end responsible? Where is the room for improvement?
And that's a very good question. I think that's something we could actually all reflect on for quite a while. So René, you raised your hand. Go ahead.
Yeah, I think it's a very much a joint responsibility. It's a responsibility partly for us as a scientific community to be transparent on the things that we do and why we do certain things. It's also a responsibility of our education system. Kids go to school, they have all kinds of programs with which they are being taught certain things. I think this also has its place there in terms of bringing kids along and young adults along in the way genetics is playing a role in our lives in food and agriculture, just like it is important to learn them and to introduce them into other types of technologies and how they interact with these things in their lives. Well, those are only two aspects, but there are more angles to this as well, and other actors that also have a role to play in this whole story.
Thank you, René. I think Irène had raised her hand.
Yes, I think it's very important that we assume the part of joint responsibility, that we improve the end result of this communication, but that we are not naive on politicians. Because when we were talking the question before, in fact, genome editing are like small farmers in the agricultural common policy. It's something that it's easy to be for or against. So everybody wants to defend small farmers, and everybody is against the Frankenstein of agriculture. And we even have a very strong text. And the groups, the political groups that are against, they are on a no. That is not a no, that is a science-based. It's a political marker for them. They know that they do not have the majority, but they can create a good face relationship with the people who vote for them. And why do I give the example of the CAP? Because what happened to the other groups at the vote of the CAP, we had more than 5,000 emails telling us, you are bad MEPs, you should defend small agriculture. And I got quite furious with one group, because I told them, in fact, this new CAP is really very protective of small farmers, and you wanted 10 points, and we deliver on 8 of them, and the 2 that were not delivered had nothing linked with the size of the farm. So I think that we should not be naive. Let's talk direct to consumers. Let's explain the consumers, they have brains, and the politicians, they want to be elected. So I think that even we can try to make the Greens change their mind, but maybe we should talk to the people who vote for the Greens and who want a reduction of pesticides, to tell them that it will not come from the sky alone. We will need to change something, and that are the different choices. And with the choices that are with the impact measurement, because nobody in Europe wants to spend €1 more for their food. So it's of course we can stop using tractors and use horses, and the food will be increased a lot. So for genome editing, we need to really share the responsibility, but not be naive, and maybe try to talk direct to consumers.
Thank you, Irène. I agree. I think it's important to consider that politicians have to answer to the base that elect them. Max, go ahead.
Yes, I just would like to add that absolutely it's a joint responsibility. And I think that what we need to do is also be able jointly to agree the story that we're going to tell about this. So we need the scientists behind it. We need our extremely good research side behind this. We need our politicians behind this, but we need also all the farmers to tell how we do, because it's not only gene editing that the people today are quite far away from. They don't really understand today's agriculture even. So I think we have here quite a big job as described by Irène, but I think it's a joint effort, and I think that we have started it today here.
I agree. I think it's very important that we're all promoting the same message and that we show that it's a united message as well. Does anybody have anything else to add on this? No? Nick, then we can move on to the next question.
Yeah, there is another question. It's related also to the current pandemic we are, and it's actually directed to Ms. Irène Tolleret. So like for COVID-19, the European Parliament voted for speeding up approval of the vaccine research. So it shows there is a clear sentiment to and belief to new safety of technologies. And so concrete, the question is, what do you think, would the European Parliament also vote for a speed track of genome editing regulations to save, for example, the vineyards in Europe if they would be under a huge threat?
I'm afraid that at the moment, the answer is no. I think that I was very hopeful that the vaccine for COVID could come from genome editing or something like that, and so that we had this subject on the table. But at the moment, I think the answer is no. So we really need to discuss the subject. Because some things we will need to not authorize them, especially when you see the creativity that some guys have in China. It's very important that we discuss what we do, but we also discuss what we don't do, and in a European. But at the moment, I don't see any possibility to accelerate that through a risk like that.
Maybe a comment to that as well. Perhaps indeed the current vaccines that are coming up on the market are not gene edited products, but all of the things that are coming on the market are heavily technology related, and most of them involve an awful lot of genetic engineering. So a number of the vaccines that are going to be introduced onto the market soon for COVID-19 are live genetically modified organisms.
Thank you. Does anybody have want to reflect on this? No? Then we can move on to the next question. Go ahead, Nick.
Yes, there's another question. I think that this is very relevant for Holger, because he was, let's say, someone of the founding fathers involved in bringing genome editing to plants, and particularly in Arabidopsis, as from personal experience. And related to that is like, how do you think, Holger, that like patenting and intellectual property will have an impact on the use of this technology for research institutes and small companies?
Well, this is, of course, a very good question. This is always a question we have to address. And of course, the patent situation is not so easy if you see the current developments. But I just want to give you one example that might also help to solve this problem to a certain extent. There are certain Cas nucleases that more or less can be freely used. That's at least a company in the States is advocating this. And at the moment, the German Breeder Society is thinking about setting up a program where they also discuss with us whether we cannot adopt these kind of nuclease also for using in breeding to avoid these problems that might arise with patents. But there's, of course, a very general thing that's going on in the field, and you always have to be aware of this. Of course, there is a lot of money in the field, especially in the medical research, if it comes to gene editing. But this also has the consequence that more and more companies are looking for alternative enzymes, and we see more and more patents coming up. And this also means that in the end, the costs to use one or the other enzyme will be reduced over time.
Thank you. Does anybody have anything to add to intellectual property related to gene editing? We'll stay away from that. Or no, René, go ahead.
Well, I agree with Holger in the sense that patenting in the gene editing area may create somewhat of a threshold, but the fact that the different alternatives are there and being developed will have an impact on that threshold cannot become too high. So compared to the threshold that the GMO regulation represents today, which is in Europe an unsurmountable threshold, I think the patent landscape will create somewhat of a threshold, but I think it will not prevent small companies or medium companies making having the ability to use this. And another big difference is that when you talk about patents in this area, that is something that we're confronted with on a global scale. So everybody in different parts in the world is being confronted with that. So there, all over the world, we have an even a level playing field, where in the legislation, there is a difference between Europe and the rest of the world.
Yeah, if I can jump in here as well, I mean, from the farmer's perspective, we are not plant breeders and we are not trying to patent what we do either today. We just hope that there will not be too much possibilities for these patents so that it will start to strangle the small and the medium-sized plant breeders in Europe. We are working in a different way here than what, for instance, over the pond in the USA. So we are basing our plant breeding very regionally on small and medium-sized plant breeders. And for that, it could be a little bit of a struggle if you would have a lot of the plant or some of the genes patented. So I hope that we will see a more open way of doing it.
Thank you for that, Max. Irène?
I will just jump in and make the link with the question before. This is a political key vote because for the groups that are against genome editing, one of their arguments is that nature is not patternable. And we have seen cases of exaggeration. When you have the small producer in South America that has a problem with his seeds from so we consumers worldwide, they have heard about this. So maybe they have not heard about CRISPR, but they have heard about the fight for seeds. So I think that maybe a deal should be made on that. If we want to accelerate the legislation to facilitate genome editing, then maybe all the people involved in research and the industry need to come with propositions on non-patterns on some sorts of cultivars, like very sensitive food, things like that. Because if we have an agriculture that is using non-pattern seeds, and that because we develop genome editing, we reduce pesticides and we reach the goals of sustainability, but we privatize totally seeds, there is a discrepancy in the speech. So I think that really the subject of patterns needs to be put on the table and discussed with all the stakeholders.
I agree. I think it's something that really needs to be discussed because it's a very contentious issue on a lot of points. So we're almost out of time. Can we have just one last question and then we will wrap up?
Okay, then I will try to make it also more general. It's actually directed to Mr. Schulman. So as a farmer, you can choose different seed varieties. We have different practices such as organic farming. And the question is related to like, now it's shown like exclusion, rather one or the other, but there should be freedom of choice. So the question is, how do you see as a farmer that like genome editing of varieties and let's say varieties that are used for organic cultivation can be cultivated in Europe?
Yeah, I mean, coexistence in between today, like you would say, the conventional farming and the organic farming, it works today. I also see that with these new type of varieties that it looks like that we can do through genome editing actually would even benefit even more the organic side of today than what really would come in then to the conventional side. For sure, with the legislational change now, even the conventional side will get more benefits out of it, more pest tolerant, more drought tolerant, and also more disease tolerant varieties. So I just think that what we need to do is to have a good coexistence legislation in place, if nothing else. But then one more time go into this kind of a labeling and give the choice, give a possibility for the people to choose what they grow, but also what they eat. So I think that this is the main thing. I think that if we start to put by someone, if it is the legislators or through the pressure of some parts of the society, people into boxes and just say that you can only do this and that, that will not work in the long run. I think that we will use all of these new technologies mixed. For sure, organic has its own rulings, but I also have a feeling that there we will also see that they will also change. I mean, new varieties are coming in there as well. How will they be then developed? That we for sure don't know today, but I would say that there will be development going on. Which tools will be used there? I think that we are talking most probably about one, but when it will be available for that side, it might take much longer time. But I think we need coexistence legislation if necessary. But otherwise, I think we have to rely on the consumer's choice. They have to be able to choose. So we have to give them also some possibility to take their own decision. It's not an easy thing. I know. I get a lot of questions coming into this as well, how to do it. And I think that we are not totally yet there that we can say, let's try. But I would like to be able to say, let's set up a trial thing. Today, I have neighbors growing organic next to my fields. I'm conventional, and I'm also using quite a lot of organic ways of also planting, keeping the crops in the rotation, etc. So we are mixing these two systems already. Why not mix a third one?
Thank you for that. Does anybody want to reflect on that before we wrap up? No? Okay, well, thank everyone for your presentations and your contributions on the panel and replying to the answer or questions. I will now give the floor to Mark Cornelissen. So he works at BSF as head of seeds and traits, and has also acted in the past years as the chair of the board of Plants for the Future ETP. So Mark will wrap up a little bit, try to summarize. Go ahead, Mark.
Thank you, Amrit. I think we had a very rich, fantastic discussion actually, where we touched on all the different elements that relate to gene editing. But I would like actually probably to review a few points that I noted. But just to be sure, I'm not the head of seeds and traits of BSF. I'm just the lead for sustainability. Well, first of all, I think what we touched upon is that gene editing is subject of a very, very contrasting discussion. It's about communication. It's rational versus emotional. It's to some extent even, how do you call it, tech push versus market pull. But then in this case, there is a lack of market pull. And the result actually is that there is not a common picture in the discussion and there is not a common frame. And so advancements actually need a change in this initial setting. And it's more than trust. We touched upon it. It's also actually a common communication. But what I above all think, if you want to have market pull, it's important not to think about societal benefits, but the benefit to the individual. You have really to ask the question, why would an individual adopt innovation? Because that by definition goes together with uncertainty. We also touched upon the fact that there's a lot of backwinds actually that help us in this discussion. The unfortunate global challenges linked to sustainability, sustainable production, the repair of the ecosystem we touched upon, climate change, healthy diet. And at the same time, I think also we heard there's an enormous inconsistency in Europe linked to the Green Deal, farm to fork strategy, and the approach to gene editing. That actually is a kind of strange situation where on the one hand you see that there's an enormous knowledge base being developed that could help progression, and we hamper. And that what came also out of the word cloud. I mean, frustration, lack of support. And the question is, how can we capitalize on this knowledge, still serve the global challenges, stay internationally competitive at the different levels in the value chain, and do we fully appreciate and communicate and understand ourselves what would be impacts of a plan B that we don't have it today, I think very clearly. Then I thought that Holger gave a beautiful example that the discussion has moved to the wrong end. I mean, gene editing is a surgical change, and that happens below background levels of mutation rates. So why is the discussion running on the process, and why is it not running on the product? Because that's the only thing that remains after you realize this mutation rate. And so what I thought was a highlight in discussion, we touched upon the fact what would need to happen before people actually align on this topic. And the COVID example came up, I think it's a great example, because there suddenly the world runs without water, and then people suddenly wonder, how do I get water? What is necessary to happen that we put our shoulders on the same thing? And I think from the discussion of today, the reluctance also to put in the word cloud immediately, these are the flagship opportunities, these are the success stories. It's very difficult to communicate on the relevance of what is at the table. And I think today the Green Deal, the farm to fork strategies, these are very good aspirational goals. But what is lacking, I think, to my opinion, is an actionable roadmap for all of us in the value chain, so that we can talk about expected impacts and have a similar kind of communication strategy, as Max said so very well. So that were my main observations during this seminar.
Thank you very much for that, Mark. So thank you again to all our speakers, our panelists, and Mark for wrapping up. And thank you all for the participants for joining and adding your thoughts to the word clouds and the polls and so on. So I hope this is the beginning of more discussions. We hope to have more of these events and include more people and have more topics. It's definitely something that should be talked about more and more. We need to open also the conversation to a wider audience. I think it's important to reach out as well to society, citizens, regular people who are wondering why do we need genome editing at all in Europe, because there is food security. So I think these are things that really need to be discussed. So thank you everyone for today, and we look forward to seeing you all again.
Thank you.
Thank you very much. Bye-bye.
Bye-bye. Thank you.